Hi, Ethan. You're the only one who's shown up so far. Good morning, folks. Welcome to the biology review session. Does anybody have something they want me to go over? Any specific questions you guys have? Okay, well, what we'll do then is I'll go over some of the parts of the class that I know from experience students tend to struggle with. Feel free to interrupt me at any point. You can raise your hand, you can chat, you can unmute and talk to me, whatever. I can handle being interrupted. It's not a big deal. So if we're going to review some of the stuff, we're going to start at the beginning of the semester. And, oh, I see somebody's in the chat. One of the problems a lot of people have is the difference between a hypothesis and a theory. So in science, a hypothesis is an educated guess. It's what do you think is going to happen? A theory is a well-supported explanation of what happens and why it happens. So when we talk about the theory of evolution, we have lots and lots of evidence supporting the idea that evolution happens, and we can explain a lot of the reasons why it happens. Why do peacock males have these huge tails that make it difficult for them to fly and escape predators? The reason is because they use those tails to communicate to the female, hey, look at me. I am such a badass peacock that I can avoid predators, even with this big tail that, you know, kind of slows me down and makes it hard for me to fly. So we have an understanding of why a lot of these things happen. Now, in science, theories are constantly being updated, tested, and so we talk about why the peacock has that massive tail to attract females. That doesn't mean we understand everything about evolution. And people are still researching it, trying to figure out Well, in this situation, why does this plant or animal, why did they evolve this way? So when somebody figures something out, when a scientist figures something new out in the theory of evolution, it just gets added to the theory. And in science, we don't like to say anything is proven or certain, but we can say something is disproved. So we can say that is not true. There was a hypothesis early in the history of HIV, early in the HIV pandemic, that said HIV got into people because of the polio vaccine. Yeah, Ethan, go ahead. Ethan, I can't hear you. So either type your question in the chat or unmute yourself. Yes, there is a study guide. Let me go to the my Canvas page. I'm just doing this behind the scenes. It is in ooh. I actually don't see that showing up for you guys. Okay, I will put it in this week's module as soon as we're done with the review session. Good catch, Ethan. All right, going back to the whiteboard. So there was an idea that HIV got from chimpanzees into humans because of early testing of polio vaccine on people in the Congo region of Africa. And there are still people who believe that to this day, that it was because of the polio testing. Well, there are two problems with that. We can disprove that because we have tissue samples of people who, and their tissue samples contain HIV, but the tissue samples were taken before the polio vaccine was tested. So we can disprove that idea. So science is all about doing experiments. And there are certainly observational experiments, but when we think about these, we tend to think about laboratory experiments. And in a laboratory setting or in medicine, we are going to have two groups. And these can be groups of cells, groups of plants, groups of people. The control does not get treatment. Whatever you're looking at, if you're looking at a drug, the control group does not get the drug. If you're looking at the effect of high phosphorus in the soil on plant growth, The control group gets soil that has normal levels of phosphorus. So this is just the normal group. The experimental group is the one that gets the treatment or the drug. Very, very important. Only one thing should vary between the control and experimental groups. So if you're looking at phosphorus levels in the soil and its effect on plant growth, you want the plants to get the same amount of sunshine, the same amount of water, be at the same temperature. If you're looking at the effects of a medication on people, you want them to have this, be the same age, have the same health background, eat similar diets. And all of that stuff is important. All of those things, it's the same between the groups, are the control variables. Things that could vary between the groups, but we're making sure that they don't. What varies is the independent variable. So the IV, sometimes abbreviated IV, is, in the plant example, the amount of phosphorus in the soil. What you are measuring is the dependent variable, or the DV, because it depends on whether that group got the treatment that is the independent variable or did not get the treatment that is the independent variable. Are there any questions on hypothesis theory and how experiments are set up? When I was grading your assignments, most of you guys, a lot of you guys got this. It seemed to work, but some people got very confused about the independent and dependent variable. And several people did not understand control variables. So I wanted to make sure you guys get that. All right, I'm going to clear this screen. The next major topic is what is required for us to say something is living. And we have heredity. They produce offspring that look and behave similar to the parents. We have evolution, which is a change in the species. We have adaptability, which is kind of another way of saying evolution. So we'll just skip that part. Responsiveness. They respond to changes in the environment. There's at least one cell. They have metabolism. They grow and develop. They have homeostasis. So these are the characteristics that we're talking about here. Evolution is being able to have offspring. Growth and development, the offspring get larger and may change appearance or behaviors as they grow up. This is more true in multicellular organisms. I mean, we look at a baby and we think it looks like an adult, but if we were aliens and we came to this planet and we saw babies and adult humans, we might not recognize that those are the same species. Babies are kind of rounder and softer and unable to take care of themselves, whereas adults, you know, we're taller, we've got long limbs, and of course most of us can take care of ourselves. Metabolism is energy and manufacturing. When I say manufacturing, we're really talking about the biological or macro molecules. So this is a good question. That's a good thing to know. It's a very good thing to know. People often just assume that metabolism refers to energy. And homeostasis, the maintaining internal conditions at or near optimum. That's one way of putting it. There's several different ways of defining this. You can also say maintaining equilibrium. Equilibrium means that you might go up and down from a set point, but the average stays stable. This concept here, this near optimum idea, is important. So you're maintaining conditions that are going to make your cells or your body most efficient. And if you get out of that equilibrium, if you lose that homeostasis, that is usually a disease state. So if you can't control your blood sugar, that's diabetes. If you can't control your body temperature, that's either a fever or hypothermia. Pretty much everything in your body has to be maintained at or near a specific set point. And so homeostasis, a word we're not all that familiar with. We don't use it in society very much. It's another word that's very important for you to know. Questions on this one? All right, I'm getting ready to erase this. Oops. Okay, chemistry. So the definition of an element is all atoms have the same number of protons. So every atom of hydrogen has 1 proton. Every atom of helium has 2 protons. Always, always, always. When we talk about these subatomic particles, we have protons, we have electrons, we have neutrons. Let me see if I can do this in this space. Protons are in the nucleus. They have a +1 charge and a mass of 1 atomic mass unit. Electrons orbit the nucleus, have a -1 charge. Their mass is so close to 0 that at this level of understanding, we just consider it to be 0. And neutrons are in the nucleus. They have no charge on them. And their mass is 1 atomic mass unit. So if I told you there was a hydrogen atom with a mass of 1 and no charge on the atom, you should be able to tell me there's 1 proton, 1 electron. That's because there's no charge. And because the mass is 1 and the numbers of protons is 1, there are no neutrons. If instead I said a hydrogen atom with a mass of 1 and a +1 charge, we're still going to have a proton, 1 proton, always, always, always. But now, because there's a positive charge, that tells you that it is missing that electron. It has given up that electron. It is now an ion. Specifically, because it has a positive charge, it's a cation. And the mass tells you it still doesn't have a neutron. If we have a hydrogen atom with a mass of 2 and no charge, still going to have 1 proton. There's no charge, so you have to have 1 electron for each proton. But now we have a mass of 2, and the rest of that mass has to come from a neutron. This, the difference here between the number of neutrons mean it's an isotope. Isotope, there we go. A charge means there's a different number of electrons when compared to protons. And those are ions. Cations if positive charge, anion if negative charge. And some, but not all, isotopes are radioactive. I remember we had a good long discussion about that one. All right, I'm getting ready to erase this screen unless you guys have a quick question. You're all going to hate me when I'm in person and I make you talk. Solutions. So a solution is a liquid or gas mix of different molecules. I didn't go over types of bonds, did I? Okay, I'll do that in just a second. Okay, so a solution consists of a solvent, which is the majority part of the solution, and there's only one solvent. It's whatever makes up whatever is in whatever is in the most volume in that solution. Solute is one or more molecules that are the smaller part of the solution. So when I make coffee in the morning, I put water in the coffee maker. I put coffee grounds in the filter. I run it. The coffee itself is a solution. And it's got things like caffeine and flavorings that have gotten into that solution. But the majority of it is water. So water is the solvent, and that caffeine, I add a spoonful of sugar, and the flavorings, those are all solutes in the solution. And a molecule is two or more atoms with a bond. They've got a bond between them holding them together. And we talked about 4 types of bonds. Ionic is when you have 2 or more ions. So you have a cation and an anion, one with a positive charge, one with a negative charge, that are attracted to each other. Opposites attract in chemistry. Sometimes you can have 2 atoms that share electrons. Those are covalent bonds. Think about CO. You think about things like coordinate, cohabitate. That CO tells you they're working together. Oops. And if it's polar covalent, they're sharing the electrons unequally. So one of them kind of pulls harder on the electron than the other. Non-polar covalent, they're sharing electrons equally. With a polar covalent bond, you have partial charges on the molecule. So it's not a +1 or a -1. It's much, much smaller than that. But that's important because of hydrogen bonds. And this is two polar covalent molecules that are attracted to each other. One is partially positive, the other is partially negative. With hydrogen bonds, they're pretty weak. But large numbers of them work together and are very, very important in macromolecules and in water. So these types of bonds are important that you have at least a basic understanding of what they are. If you go on and you take chemistry or more advanced biology classes, they're going to assume you know these. And I'm giving you a second in case anybody has any questions. So when we use the terms polar and nonpolar, polar molecules are hydrophilic. So those two terms are often used interchangeably. Hydrophilic means water loving. Nonpolar molecules are hydrophobic or water hating. What this means is that in a cell, if you have a molecule that is hydrophobic, it's not going to be floating in the cytosol, in the liquidy part of the cell. It's going to be most likely in a membrane because the interior of those membranes are also hydrophobic. And so if you make a protein that's hydrophobic, it's most likely going to be in a membrane. If you make a polar molecule, it will be in the liquid part. It's not going to be in the membrane. It's going to be in the cytoplasm or cytosol. So this can determine where, oops, a molecule can be found in a cell. Organic molecules refer to any molecule that has at least one carbon and one hydrogen atom. So we don't include carbon dioxide, even though it has carbon. But if we have something like ethanol, which is an alcohol, it's got a carbon, it's got at least one hydrogen, and so that's going to be considered organic. And these tend to behave in similar ways. So we can look at organic molecules and get an idea of how they're going to act. Now, the bigger the molecule, the harder this is, but small molecules, we can make some predictions on how they're going to interact with other molecules, where they're going to be found in the cell, and that kind of stuff. And inorganic molecules, molecules that don't have carbon or have carbon but no hydrogens, those behave very differently. All right. The biological molecules. There are four major categories here. Proteins, which are made up of amino acids. Carbohydrates, that are made up of simple sugars. Nucleic acids, that are made up of nucleotides. And lipids, which have a lot of variability to them. So they're not just strings of monomers like proteins and carbohydrates and nucleic acids. They're going to have different structures that can be, in fact, highly variable. But what they do have in common is they are going to be hydrophobic. Proteins, carbohydrates, nucleic acids, it depends on the individual molecule. The protein, if it's going to be hydrophobic or hydrophilic, it depends on which amino acids are in it. Carbohydrates, generally they're hydrophobic, but not all of them. And nucleic acids tend to be hydrophilic. Proteins have a lot of different functions in the cell. So we can think of them as enzymes that speed up chemical reactions in the cell. You can also say they do the work of the cell. They're also involved in movement. Support, structure, all kinds of stuff. Carbohydrates are very important for energy metabolism. They also have some role in structure, but energy metabolism is what we primarily think about with those guys. Nucleic acid tends to involve information, storing information or moving information around the cell. And lipids are highly variable. I'm not gonna try and give you a simple description of that. All right. We talked about cell theory. All living things are made of one or more cells. The smallest thing considered alive is a single cell. And cells come from pre-existing cells. This is a pretty simple thing to remember, but it's important. There's the endosymbiotic theory, which talks about the origin of eukaryotic cells, the origin of mitochondria, and the origin of chloroplasts. And this states that a prokaryotic cell and it would be in the group known as archaea. We didn't really talk about this in class. Engulfed a bacteria cell, which is also prokaryotic, but didn't digest it. Instead, it formed a symbiotic relationship. The archaea cell provided nutrients, protection, and the bacteria evolved into the mitochondria, which produces more energy per food molecule than the archaea cell originally could. So there was now an excess of cellular energy. And remember, cellular energy is in the form of the molecule ATP. And so that is how the eukaryotic cell began. You need a lot of energy to make membranes. So if you want a membrane around your nucleus, you need extra energy. If you want specialized organelles that are more efficient and allow for distinct chemistry inside of them, like different pHs, different salt concentrations. You've got to make membranes, which takes ATP. And the chloroplast formed when an early eukaryotic cell engulfed a cyanobacteria. That's a bacteria that can do photosynthesis, and et cetera, et cetera, et cetera. Formed another symbiotic relationship there. And the chloroplast is where photosynthesis occurs in modern day plants. How's everybody doing out there? Pretty good. Okay. It's amazing how fast we can get through several weeks worth of lecture when we're just reviewing. All right. We didn't talk yet about pH, acids, bases, and buffers. So let's do that now. pH is a measure of hydrogen ions in a solution. So these are hydrogen atoms that have a positive charge on them. An acid is any molecule that increases the concentration of hydrogen ions in that solution. And a base is any molecule that reduces the concentration of hydrogen ions in that solution. So all proteins have a specific pH where they function best. If they get out of pH range, if the pH is too extreme, the protein can denature. It can break down and not function. So our body controls pH very, very carefully. The pH in your stomach is designed to help kill bacteria that you might ingest, that you might bring in with food. When the food goes into your small intestine, that has a different pH that is optimum for the molecules, the enzymes that are involved in breaking food down so that you can absorb the nutrients you need from it. Your blood has a very specific pH at which those proteins work. And all of these pHs can be slightly different. The pH scale is a log scale, which is hard to grasp. But what that means is a pH of 4 will have 10 times concentration of hydrogen ions as a pH of 5. And on the pH scale, neutral is 7. Acidic is less than 7. Basic or alkaline is more than 7. So one of the homework questions, one of the options was talking about seeing if alkaline water would change your body's pH. You don't want that. It doesn't. If it did, it could kill you. If your pH gets out of balance, if it's not in the right range, that can be fatal. And in some places like blood, that pH, the range that the blood can function at is a very, very, very small range. You really don't want that to change at all. Buffers, that's right. Buffers are molecules that help maintain the pH of a solution. So you've got buffers in your blood that help maintain your blood pH right around 7.2. You've got buffers in your small intestine that maintain the pH there. That's also somewhere in the low to mid 7 range. You've got buffers in your stomach that help maintain the pH there, right around like 2. So it's very, very acidic in your stomach. So buffers are critically important for maintaining the pH homeostasis, which is critical for functioning of your proteins. Okay, we've got 5 more minutes. Does anybody have a request? No requests. Okay. All right. Can I explain control variables again? Absolutely. Absolutely. So in experimental design, control variables are everything that is the same between the control group and the experimental group. So this is easy to picture with medical drug testing. So if we're looking at a new drug and we're trying to figure out if it's effective, you want to make sure that both groups should have people the same age, a mix of sexes, same level of health. They all have, if you're looking at heart disease, you want everyone to have the same degree of heart disease or the same type of heart disease, because some people will have more severe heart disease than others. Same exercise habits, similar eating habits. So you don't need everybody to be on the exact same diet, but if you have vegetarians, vegans, and omnivore diets, you want to make sure that the control group has roughly the same percentage of vegetarians as the experimental group. The control group has roughly the same percentage of vegans as the experimental group. You don't want all of the vegetarians and vegans in one group and all of the omnivores in another, because diet can have an effect. And so that's what we mean when we say control variables. All the things that you're holding the same, that you're making sure do not vary between the groups. Because what you want to vary are the independent variable, in this example, the drug or the treatment, and the dependent variable, which is the result. Experimental is the one that is being treated with the drug or a different treatment. So if you want to know if a drug works, the experimental group gets the drug. The control group is the one that you are comparing the experimental group to. That's a good question. So if you just have an experimental group, you got a bunch of people who have allergies because the Santa Ana winds are blowing, and you've got a new drug that you think is going to, really clear up their sinuses and make their, make them less snuffly when the Santa Anas blow. You can't just get a group of people and give them all the drug and see what happens, because you don't know what would have happened without the drug. So you need a group that gets the drug and a group that doesn't get the drug, so you can see how the drug changes the results. Does that clear that up for you guys? Good, glad to hear it. All right, we are almost out of time. Hold on just a second.